Hello students, welcome back. Welcome back to my biology online classes. In my previous all lectures, you have already known that I have started cell signaling, signal transduction, and in my previous classes I have discussed with you about G protein coupled receptor and different types of secondary messenger. Protein kinase, which one is very much important. As well as protein kinase, another term is very much important to describe cell signaling or signal transduction. The two terms is protein kinases and protein phosphatase. As here AS is added, so we have a clear conception that it acts as enzymes obviously, and they are protein in nature as it indicates directly. Protein kinases and protein phosphatase. Kinase, when kinase this term is used, means this is related to somehow this is related with kinetic energy and it needs ATP to do its work, and phosphatase means somehow phosphate ion is involved. Now let's have a look clearly what is protein kinases and protein phosphatases. For activation of all cell surface receptor. Cell surface receptor means we have this knowledge that in case of cell surface receptor, the receptor is present outside the plasma membrane, and its transmembrane receptor means some part of this receptor is outside of the cell, outside of plasma membrane, and some part resting, some part inner part is inside, touch inside the cytosol. So they are all transmembrane in nature. Activation of all cell surface receptors leads to change in protein phosphorylation through the activation of protein kinase. So cell surface receptor works with the help of protein kinase and change in protein phosphorylation with the help of protein kinase. There are many types of protein kinases involve a signal transduction. They all have the same basic catalytic activity. It has catalytic activity as it acts as enzyme. What is their working principle? They add a phosphate group. Phosphate protein kinase. Protein kinase add a phosphate group to an amino acid in target protein. Add a protein kinase working principle is that they add a phosphate group to an amino acid in target protein. The phosphate is provided by ATP. The phosphate group is provided by ATP. All of you know in triphosphate. Here triphosphate are there and the terminal phosphate is released. Okay, and to do this reaction energy is also released. And this protein kinase is classified by the types of this amino acid that they phosphorylate in the target protein. Let's have a look about the classification of protein kinase enzyme. 5 groups of protein kinases are there. Protein, serine, threonine kinase. Here they can phosphorylate either serine or threonine, and all of we know about serine, threonine. There are 21 essential amino acid inside our body. Serine and threonine are one of them. Protein tyrosine kinases phosphorylate tyrosine in the target protein residues. Dual specificity kinases. So in this case phosphorylate either serine, threonine or tyrosine in the target protein residues. Histidine kinase. Here in case of histidine kinases phosphorylate target proteins on histidine residues. Aspartate kinases phosphorylate target proteins in aspartate residues. These are the 5 types of protein kinases, and these protein kinases may be membrane bound or cytosolic on the basis of their location inside the cell. So this all about protein kinases. Now come to the next point, protein phosphatase. Activity of protein kinase is opposed by, mind it very carefully, is opposed by protein phosphatases, and this protein phosphatases remove phosphate group. Kinase add phosphate group and phosphatase remove phosphate group from specific target proteins, and depending upon structural parameters like protein kinases, this protein phosphatase also can be classified. Remove phosphate group, serine, threonine, specific phosphatase. Point number 2, phosphotyrosine phosphatase, threonine, tyrosine, prokaryotic, not eukaryotic, mind it carefully, phosphoaspartate phosphatases. There are 4 types of phosphatases: serine, threonine phosphatases, phosphotyrosine phosphatases, threonine tyrosine specific phosphatases, prokaryotic phosphohistidine, and phosphoaspartate phosphatases. Now let's concentrate about some basic informations which will help us to better understand signal transduction. By hydrolyzing phosphoric acid monoesters into a phosphate ion and a molecule with a free hydroxyl group. So this is working principle of phosphatase. It remove phosphate group but how? By hydrolyzing phosphoric acid monoester into a phosphate ion. Okay, hydrolyzing phosphoric acid monoester into phosphoric ion and a molecule with free hydroxyl group. And another enzyme, phosphodiesterase, enzyme that break a phosphodiester bond, kinase enzyme. This is also called as phosphotransferase, as it transfer phosphate group from a high energy donor molecule such as ATP to substrate. Phosphorylase enzyme, what can they do? Enzyme that catalyzes the addition of an inorganic phosphate to an accept. Come to another very important interesting point, agonist and antagonist. What are agonist? Agonists are those molecules that have resemble with signaling molecule, that can mimic signaling molecule. They are structural analog which mimic the function of signaling molecule by binding to its receptor and causing the normal response. This one is called agonist. Another term is antagonist. These are also structural analog which bind to the receptor and but induces no response. This induces no response. So these are some important whatever you can say, some related term of signal transduction.